In 1952, two men called Hershey and Chase discovered that DNA was a genetic material. DNA stands for deoxyribonucleic acid. It is a molecule composed of two chains that coil around each other to form a double helix or spiral staircase shape that carries genetic instructions used in growth, reproduction, and more. Rosalind Franklin used X-ray technology to develop pictures of DNA that helped determine the structure. Two men, Watson and Crick, then used Rosalind's pictures to discover the DNA was in the double helix shape.
The building blocks of DNA are nucleotides, four nucleic acid units that make up DNA: phosphate group, deoxyribose sugar, and nitrogen-containing base, single or double ring. The four nitrogen bases found in DNA are adenine, A, guanine, G, thymine, T, and cytosine, C. Hydrogen bonds form between the nitrogen bases, causing DNA to twist. Adenine and guanine are double ring purine. Thymine and cytosine are single ring pyrimidines. A scientist named Chargaff discovered the base pairing rules that A pairs with T and C pairs with G in DNA.
RNA and DNA have some differences. In RNA, instead of using thymine, you use uracil. RNA is just a ribonucleic acid. It contains ribose sugar instead of deoxyribose. Also, RNA is a single-stranded and DNA is double-stranded. The DNA first goes through transcription, where part of the DNA is rewritten into a strand of messenger RNA, or mRNA, inside of the nucleus. Step 1 of transcription is when RNA polymerase attaches to the DNA and unwinds the DNA and breaks the hydrogen bonds. Next, RNA polymerase adds new bases to the mRNA strand using the different base pairing rules, where uracil pairs with adenine. RNA polymerase then rewinds the DNA when it is done. Next, the RNA strand moves out of the nucleus to the ribosomes, which make protein for translation. When transcription is finished, you get an mRNA strand that will be used to make a protein in the cytoplasm. The mRNA strand goes to translation, where it is decoded into proteins with the ribosomes. These proteins are molecules made up of amino acids. A codon is 3 nitrogen bases in mRNA, which code for a specific amino acid. Every codon is read and matched with a tRNA. Amino acids link up and make a protein.
During digestion, the sight, smell, or taste of food stimulates the release of digestive enzymes by salivary glands in the mouth. The mouth is the beginning process of mechanical digestion. When the food is chewed, the tongue mixes saliva in with the food, and once it's moistened and swallowed, it passes into the pharynx. From the pharynx, the food moves to the esophagus. It passes the food from the pharynx to the stomach. The stomach digests food both chemically and mechanically. Hydrochloric acid kills bacteria and gives the stomach the low acidity needed by digestive enzymes that work in the stomach. Pepsin, the main enzyme, chemically digests protein. All absorption takes place in the small intestine. Trypsin and peptase digest the proteins. Amino acids get absorbed into the bloodstream. Once the amino acids are in the bloodstream, they diffuse into the cell and attach to their proper DNA. During transcription, the DNA directions for hair cells are transferred to mRNA. RNA polymerase uses base pairing rules to create mRNA. This all takes place in the nucleus. During translation, mRNA is decoded, and every codon is attached with the tRNA anticodon. The amino acids then link up and become a protein.